Good morning. I would like to call to order the ICER Midwest CEPAC meeting on the treatments for spinal muscular atrophy. At the beginning of every Midwest CEPAC meeting, we want to ensure that all speakers disclose any financial relationships with industry. Every member of our Midwest CEPAC panel who's present today has met the ICER conflict of interest policy. I will call on each of you to please introduce yourselves, your work, and note whether you have any updates to this declaration. I'll start. My name is Jill Johnson. I'm the current chair of the Midwest CEPAC. I'm a professor at the University of Arkansas for Medical Sciences College of Pharmacy, and I do not have any updates to my current conflict of interest statement. Okay, Bijan?
Yeah, hi, good morning. I am Bijan Bora. I am a professor of health services research at the Mayo Clinic College of Medicine and Science, and I don't have any update to my declared conflict of interest. Thank you.
Bradley?
Hey, good morning, everyone. I'm Brad Martin, professor at the University of Arkansas for Medical Sciences, a colleague of Jill's, and I have no updates to my conflicts previously reported.
Kurt?
I'm Kurt Vandenbosch. I'm a pharmacist with the St. Luke's Health System in Idaho, and I have, excuse me, I have no changes to my conflicts of interest. Thank you.
Timothy?
Good morning. I'm Timothy McBride. I'm the Bernard Becker Professor at the School of Public Health in Washington University and co-director of our Center for Health Services, Economics, and Policy Research, CASPER, as we like to call it. Thanks.
Thank you. Don?
Morning, I'm Don Casey, Associate Professor of Internal Medicine at Rush Medical College and also Adjunct Professor of Quality and Population Health at Thomas Jefferson. No update and no conflict of interest or relationship with industry for this discussion. Thank you.
Thank you. Reem?
Hi, everyone. I'm Reem Mustafa. I'm a professor of medicine at the University of Kansas Medical Center, and I also direct the Evidence-Based and Impact Center here. And I have no updates to my conflict of interest. Thank you.
Thank you. Sneha?
Hi, I'm Sneha. I'm Executive Director at Generation Patient, an organization representing young adults with chronic and rare conditions. I am also a patient, and I have no updates to my conflicts of interest.
Thank you. Stacy?
Good morning. My name is Stacy Doucette-Zena, and I'm a professor of health policy at Vanderbilt University Medical Center, and I have no updates to my conflicts of interest.
Rachel?
I'm Rachel Sachs. I'm a law professor at Washington University in St. Louis, and I have no updates to my conflicts of interest.
Thank you. Jayani?
Hi, I'm Jani Jawadana, an associate professor in the College of Public Health at University of Kentucky. I have no updates to my conflicts of interest.
Okay. Stuart?
Good morning, everyone. I am a clinically retired cardiac electrophysiologist and current lead patient experience consultant for a large multi-specialty group, Trinity Health IHA in southeastern Michigan, and I have no updates to my conflicts of interest.
Thank you. So that is all of the members of the Midwest CEPAC. Next, I want to introduce the...
I think I need to declare my no updates. I'm David Kim.
David, sorry. I'm sorry.
Yeah, no problem. Assistant Professor of Medicine and Public Health Sciences at University of Chicago. I have no updates on conflict of interest.
Thank you. Apologies. So there are 13 of us today. I would like to now introduce the individual patient and clinical experts. I know that we have one of our clinical experts is running just a couple minutes late, so I'll get to him last. I would like for you guys to introduce yourselves and declare any potential conflicts of interest as well. First will be Giles Lomax.
Hi all, my name's Giles Lomax and I'm the Chief Exec of Spinal Muscular Atrophy UK, based across the whole of the UK. I actually have twins that have SMA type 2 as well, so have a really deep understanding of the organizational challenges and things that come through in terms of the pharma and industry and a lot of changes within the advocacy world, but also the lived experience of looking after the 7-year-old twins. As on the screen, SMA UK is a charity, and we have received just over 15% of our income from industry across 4 of those pharmaceutical companies in the last financial year.
Thank you. Thank you for being with us today. The next one is Portia Thorman.
Hello, I'm Portia Thorman. I'm Head of Advocacy and Community at SMA UK. I'm also mum to a little lad, now 8 years old, with SMA type 1. And yeah, what Giles just said, all of the above. And we have no updates to our interests.
Thank you. And thank you for being here with us today.
Pleasure.
Okay. Let's see. We will skip over Dr. Crawford for a second. He'll be here in a minute. We will go next to Hugh McMillan.
Yep.
Dr. McMillan.
Thank you. My name's Hugh McMillan. I'm a pediatric neurologist and neuromuscular specialist at CHEO, the Children's Hospital of Eastern Ontario in Ottawa, Canada. I've been the PI for more than 25 regulated trials. My conflicts of interest relevant to SMA is I have received some consulting fees, and I have been the principal investigator for clinical trials with Novartis, Roche, and Biogen. All funds for that have gone to the institution. I have no relationship with Scholar Rock, and the only potential conflict related to myostatin inhibitors is I was involved in 2 myostatin inhibitor trials for Duchenne muscular dystrophy that did not meet their primary endpoint. Thank you.
Thank you. And thank you for being here with us today. We really appreciate your input. Once Dr. Crawford gets here, I'll introduce him. I did want to be sure to say that I will be requesting that all the speakers, including the policy roundtable participants and anyone making public comments, will be asked to announce your financial relationships with industries and other potential influences on judgment. So I will call on you whenever the time comes. At this point, I would like to introduce Sarah Emond, the president and the CEO of ICER.
Jill, thank you so much. It's really wonderful to be with all of you today. I'm going to be joined with several of my colleagues throughout the day. You'll be hearing from David Rind, ICER's Chief Medical Officer, also a primary care physician who served as what we call the senior management team lead for this project. We'll be hearing from Linda Liu, who was the lead economic modeler. She's from the University of Washington, and Dr. Jeff Tice, who was the evidence author for this report and many ICER reports, who works at the University of California, San Francisco. Next slide, please. Every time we convene a public meeting, we want to begin by remembering why we're here. We're here because there are new treatments under development, in this case, for a very serious condition. We were all struck by our conversations with patients, their families, their caregivers, but this quote from a report from Cure SMA really struck many of us as a reminder for why we continue to convene to talk about the comparative clinical effectiveness of new medicines. And that's because ultimately we want patients to have affordable access. This report told us that every single time I attempt to eat a bowl of soup or reach my canvas while painting a picture, I know I am getting weaker. Everyday wins would be things like improved respiratory function, skills for life improvement, like being able to pull up the blanket to cover yourself in bed, independently use the toilet, or put your own shoes on and keep up with peers without excessive fatigue. We are fortunate to have the patient perspective represented with us throughout today, but we will try to remember what we are ultimately talking about is making sure there's affordable access for new therapies for patients with spinal muscular atrophy. Next slide. One of the reasons that we exist is that there are a lot of questions that remain for decision makers, for patients, for their families, for practicing clinicians, even after FDA approval. Because when a new treatment is approved, It may be cause for celebration. An exciting new option might be available for patients with a particular condition. But it doesn't mean all of the questions that we may have about that treatment are answered. We might still have questions about the balance of risks and benefits of a new treatment. We might not have much information about knowing how a new treatment compares to what we're already doing. And we have important questions that we should be asking about prices, and those prices impacting costs for patients, the health system, and the government. And we always like to end every policy conversation that we have during these public meetings about what are we going to do to move forward? What are the research questions we need to answer? What are the policy options in front of us that would help us get to fair price and fair access for all patients? Next slide. We ask these questions about cost, not because we're a bunch of economists who care only about costs. It's because we care about the impacts that prices have on real people. And there are some examples here that are linked below. We've talked before at these meetings about the wards from Chicago who went $80,000 in debt because their twins were born premature. We talked about Sherry Foy, who had to have her colon removed, went $850,000 in debt, and had to declare bankruptcy because of a health condition. We know that Delaware last year told everyone they had to raise healthcare premiums 27%, mostly because of outpatient hospital costs and drug costs. And that the most recent report from NEBR about the impact of rising premiums showed that wages go down when health insurance premiums go up. This is why we do not shy away from conversation about cost, because on the next slide, we want to remind everyone that we can have a conversation, next slide, please, about the patients and their families in the room with the doctor making consequential decisions about treatment while still remembering everybody else in the healthcare system who is impacted by some of those decisions. We have a population health imperative in front of us to make sure that we are paying fair prices so that everyone in the healthcare system has sustainable access to high value care. Next slide. Just a little bit about the different groups and names you will hear today. ICER, the Institute for Clinical and Economic Review, is the convener of the Midwest CEPAC. The members of Midwest CEPAC are independent of ICER. They are practicing clinicians, epidemiologists, health economists, policy experts, law professors, who all care deeply about an independent evaluation of evidence and value as a way to drive policymaking. And they have my gratitude for their time they're spending with us today. Next slide. We've made everybody else talk about their financial conflicts, and so here are ours. Every entity that has given money to ICER in the last 12 months is listed on our website. You can find that full transparent list available on the website. We only use money from our philanthropic supporters, whether individual or foundations, to support our research. Any money that comes from industry is used only for our policy leadership forum, where we bring together leading pharmaceutical companies, payers, and pharmacy benefit managers to try to tackle the big issues that we're facing as a system. Our most recent report, for those who are interested, how should we pay for the GLP-1s for obesity? It's a great read. I recommend it. Next slide. A little bit about how we've gotten to this point in our process. An ICER review takes over 8 months, and that's because we have several important points of public input and engagement. We scope our review at the very beginning by talking to patients, clinical experts, the manufacturer about the key research questions we should be answering. We develop an economic model. We synthesize the evidence, and we did that in this case with our collaborators from the University of California, San Francisco, and the University of Washington. We had expert review of that draft report. That draft report went up for public comment. We revised the report and responded to those public comments. All of those response to comments are available on our website in order to produce an evidence report that forms the basis of our conversation today. After today, we will be updating that report with reflections of the votes, the deliberation, as well as the policy roundtable that we will hold at the end of today. Next slide. You'll hear us talk about value today. So just to ground those who might be new to the ICER process, when we're talking about value, we're talking about the comparative clinical effectiveness, and we're talking about how much we are spending for health gain. So the boxes here in green represent things that are typically pretty easy to measure, not always. We can know how much a patient is living longer if it's a life-limiting condition, or living better, like an impact on their ability to do things like pull up the bed cover at night or go to the toilet themselves. We can measure that. We can measure how much we're spending and saving by impacting those patient-centered outcomes. We measure all of that in our comparative effectiveness review. We never want to shy away from how important it is to remember things that are harder to measure, but are also very important when talking about comparative value. These benefits beyond health or special ethical priorities will be another key part of our deliberation today. The section of our report that reviews these was informed by multiple conversations with patients, patient advocacy organizations, and clinical experts so that we could hear about the things that matter most to them out of treatment. Next slide. Congratulations, you've made it through the opening convening of the meeting today. In just a moment, I'll be handing it over to Dr. Tice for the presentation of the clinical evidence. He'll be followed by Linda Liu, who will walk us through the economic model. We'll take public comments and have a conversation with those who are taking their time to give us feedback on our report and our deliberation. After a lunch break, the Midwest CEPAC will make votes on the comparative clinical effectiveness of several of the interventions that we'll discuss today. After another break, you'll hear from me again, and I'll be joined by patients, clinical experts, and payer representatives to say, okay, this is what the evidence is. Now, what do we do from an evidence-based policy-making perspective to ensure that we are providing affordable access to these new medicines? The Midwest CEPAC will have time to reflect as we close today's convening, and we should be done by 4 o'clock Central Time today. With that, it is my pleasure to introduce Dr. Jeffrey Tice from the University of California, San Francisco, who will walk us through the clinical evidence presentation. Dr. Tice, over to you.
Great. Thank you so much, Sarah, and thanks to everyone for being here. Next slide, please. So I just want to acknowledge my collaborators in developing the clinical evidence and comparative clinical evidence section of the report. Dimitri Nikitin is the senior research lead at ICER, and Sol Sanchez is the research assistant who worked on this, and they really did the heavy lifting. My only conflict is that my institution, UCSF, has a contract with ICER to produce these reports, and we don't have any other conflicts. I should say I'm also a primary care physician like Dr. Rind, and I do care for one patient with SMA, an adult with SMA. Next slide, please. So spinal muscular atrophy, or SMA, as most of you know, is a hereditary disease, a genetic defect in a gene called SMN1, which produces a protein that helps helps motor neurons to survive. So without that protein, there's progressive loss of motor neurons, and that in turn leads to progressive muscle weakness, challenges breathing, challenges swallowing, and challenges meeting developmental milestones as children grow. So crawling, sitting, standing. In the more severe forms, patients are unable to do any of those things. Next slide, please. Traditionally, it's been classified by SMA type, 0 through 4, and this is mostly based on age of symptom onset. There are the most severe form, you may have fetal loss, so it presents prenatally. And type 1, it presents before the age of 6 months, and so on. In this day and age where we are doing testing for this condition at birth, we no longer actually wait for patients to develop symptoms. So this old classification is no longer useful, and we focus more on the copy number of another gene called SMN2, which produces the SMN protein, but it's not a fully functional form of the protein. But when you have more of this, you have less severe forms of the disease. And you see in the old SMA types, I listed the highest motor function achieved. And when patients typically survived before we had these life-altering therapies, the most severe forms died before the age of 2, certainly. And so what we call SMN-directed therapies that really address the root cause of this disease have been transformative. And as our Chief Medical Officer, David Rind, likes to highlight, it's really one of the major medical successes of the past decade. These children who used to die can now live almost fully normal lives with the treatments that are available now. Next slide, please. With that said, so I just highlighted it used to be a fatal disease. Now, with identifying it at birth, we're able to prevent the impacts on respiratory function, so they no longer need ventilatory support, and that's life-saving. Patients wanted us to know that one of the therapies, which I'll talk about briefly on the next slide, requires a lumbar puncture, so intrathecal delivery of the drug, and that can be traumatic for young patients. And in fact, many of them require general anesthesia to receive this therapy, and it has to be done every 4 months. Among adults, we heard that gaining muscle strength is their greatest unmet need, and the new drug that we're considering today really addresses this unmet need. They also highlighted that fatigue is an important issue, particularly for older patients, and it's really not been well studied. That may reflect the changing focus as we now are keeping patients alive and now can focus on improving quality of life as well. And then patients wanted to let us know that financial toxicity is a major issue for patients and their caregivers. And it's not just because of the cost of therapies, but also the supportive things they need for home care, dealing with patients, their children who are unable to feed themselves, who have challenges breathing and all the complications that come with that. So there are lots of assistive devices and need for additional support in the home to care for their loved ones. Next slide, please. So the standard measurement, there are 3 therapies. Nusinersen or Spinraza was the first drug that became available, was approved by the FDA. It targets the SMN2 gene, as I said, so that it creates more of the functional SMN protein. And this is the therapy that has to be delivered via lumbar puncture every 4 months. It's a little more frequent initially. There's a loading dose, but then every 4 months afterwards. And that was transformative, and we gave that an A evidence rating, our highest evidence rating at the last meeting. There's also a gene therapy. I'm sorry, I can't pronounce the generic name. I'll say Zolgensma or just gene therapy. And this therapy delivers a functional copy of the SMN1 gene as a one-time IV infusion. When given early enough, and at least in the mild forms of disease, is essentially curative. And then finally, there is an oral form of therapy, Risdiplam, which is a splicing modifier, again targeting SMN2, but it increases the production of functional SMN and helps to prevent the loss of motor neurons and the progressive weakness that characterizes the disease. Next slide, please. So our scope of the review today really has 3 research questions that we're focusing on. The first is the comparative effectiveness and economic value of apitegromab, which I haven't described yet. I'll describe that on the next slide. It's the new therapy. So we want to see the net clinical benefits of that as add-on therapy to background disease-modifying therapy. And it's been studied just as add-on therapy to patients receiving nusinersen or Risdiplam. The second question that we're going to address is what is the net health benefit of Risdiplam or nusinersen as add-on therapy in patients who have already received gene therapy? In this case, we're not going to do the economic analysis. We're just focusing on the clinical net health benefit. And then finally, the third research question, is there a comparative advantage for any of these drugs when used as first-line therapy in asymptomatic infants? So again, infants identified as having the deficiency in SMN1 with newborn screening, and we're going to treat them with one of these disease-modifying therapies. Is there evidence that one of these has a comparative advantage over the others? Next slide, please. So with that, I'll turn to the new drug. This is apitegromab. It's a selective inhibitor of a myostatin precursor. Myostatin is a protein that inhibits muscle growth and strength. So if you downregulate that, hopefully we'll get greater muscle growth and strength. It is given by an IV infusion every 4 weeks. Next slide, please. The main outcome scale that's been used in all the studies is something called the Hammersmith Functional Motor Scale Expanded. It's a 33-item scale with a maximum score of 66, and the typically reported minimally clinically important difference for this scale is a 3-point change, though there's some controversy, and we'll report some other differences as we examine the evidence. Also, motor milestones are important. Can the patient sit without support? Can they stand without assistance? Can they walk alone? And so on. And finally, quality of life, mental health is a big issue in this disease, the ability to swallow, alleviation of fatigue, and improvement of muscle strength. Next slide, please. So for the first question, I'm going to describe here briefly the evidence base for apitegromab. The main trial is the SAPPHIRE trial. It's unpublished. It's supposed to be published in the next couple of days, so hopefully we'll have the final results soon. It is a phase 3 randomized trial comparing 2 doses of the drug, either 10 milligrams per kilogram or 20 milligrams per kilogram versus a placebo infusion. And these patients were between the ages of 2 and 12. They had either type 2 or type 3 SMA and were receiving either nusinersen or risdiplam as background therapy. There was an earlier dose-finding study, which I'll allude to briefly, and then there is a long-term extension, open-label extension of these studies, which is ongoing. Next slide, please. So here are the pooled SAPPHIRE trial results. So the primary outcome pre-specified by the company was pooling both the 10 and 20 milligram doses and looking at the comparison with placebo. And the primary outcome is the change in the Hammersmith scale, so that first row of results. And in the apitegromab group at 1 year, they improved by 0.6 points on the 66-point scale. Whereas the placebo group actually got worse on average, or they declined by 1.2 points. So the absolute difference between the two groups is 1.8 points. And this was statistically significant with a p-value of 0.02. And you can see the confidence interval around the estimate for the difference. For the clinically meaningful improvement of at least a 3-point improvement in the scale, a little over 30% of patients receiving apitegromab achieved that milestone compared to only 12.5% in the placebo group, an absolute difference of about 18%, and this was also statistically significant. Because of feedback, we also wanted to report just the patients who at least remained stable or improved. So you saw in the placebo group, you saw a net decline in function by the Hammersmith scale. But in the placebo group, half of patients actually remained stable or improved a little bit. In the apitegromab group, a higher proportion, so 62.7%, so almost a 13% difference, 13% more patients had remained stable or improved with apitegromab compared to placebo. And an important finding from the TOPAZ group, because these results are only 1 year, and this is a disease that patients will live with for their life. In 23 patients who've been followed for 4 years in the long-term follow-up of the TOPAZ trial. Their Hammersmith scale has remained stable over that period, so they haven't seen the decline that we saw in the placebo group here. Next slide, please. In terms of harms, there were significantly more, well, I shouldn't say significant, there were not, the p-values are not reported, but substantially more significant adverse events in the apitegromab group compared to placebo. So 19.8% versus 10%. So almost a doubling in the serious adverse events that were reported in the trial. The major components of those serious adverse events were pneumonia, 6.6% in the apitegromab group, and no patients in the placebo group, and dehydration. Now, important to note that nobody stopped therapy because of adverse events, so all patients continued for the full year. And I should say these adverse events are things you might expect in patients who are symptomatic from SMA. If you have muscle weakness, your respiratory function isn't as good, you may not take deep breaths, clear secretions, you're at a little higher risk for respiratory infections, pneumonias in particular. But it's remarkable. You would actually think the apitegromab group would have less pneumonia. So it's a surprising finding. It may just be a chance finding, but we certainly wanted to highlight those concerns. You know, again, dehydration may also represent trouble swallowing, things like that. So sort of surprising that there were more in the apitegromab group than placebo group, but again, this may be a chance finding. Next slide, please. There are a number of controversies and uncertainties. As I noted, the study had both a 10 milligram per kilogram group and a 20 milligram per kilogram group, and we didn't see a dose response here. And nominally, the 10 milligram group actually had a greater improvement in the Hammersmith scale than the 20 milligram per kilogram group. So we don't know, in part because we don't have FDA approval of the therapy yet. We don't know whether the recommendation will be for 10 or 20 or either one. So that's an important uncertainty at this point. And then the study only studied patients with type 2 or 3 SMA. We don't know in the more severe form, type 1 or type 4, less severe form, whether there are benefits. And then finally, there are no data in patients who've received gene therapy. Again, some patients with gene therapy have residual weakness and may benefit from this therapy. Next slide, please. In terms of benefits beyond health and special priorities, so things that are important for the value assessment, but not captured necessarily in the clinical data or in the economic modeling. We've heard that current therapies slow the decline, but don't restore lost function. If patients have already lost motor neurons, either in utero or before they received the current therapy, they have lost motor neurons. They have a weakness that will not improve as they get older. So there is substantial unmet need, particularly in the older populations who weren't eligible for early therapy. There are substantial unmet needs to improve strength, function, and reduce fatigue in those patients. The net clinical health benefits, as you saw, are at best small, but this may have an impact on caregiver quality of life. We saw this in the modeling of the modified societal perspective, which Dr. Liu will present. We did see significant improvements in measures of caregiver quality of life from the societal perspective. So that's important to keep in our minds when thinking about the value of this therapy. And finally, it requires an IV infusion every 4 weeks, so that requires travel to an infusion center or coordination with a home infusion program. And for many patients, this will be a burden for both the patients and their caregivers, particularly patients living far away from large healthcare centers that have infusion sites. Next slide, please. We received a number of public comments. In particular, there was criticism of the MCID for the Hammersmith. We did request additional data from the manufacturer around this. Unfortunately, they elected not to provide us any additional data, but I did try and highlight the threshold of no change, so disease stability as a response to this. And of course, we also heard that this observed incidence of pneumonia is typical to what's seen in other clinical trials. So again, that may be a chance finding. Next slide, please. So in summary, there was a small improvement in function as measured by the Hammersmith scale, so 0.6 versus a loss of -1.2 on the 66-point scale. The phase 2 study suggests stability of gains from apitegromab for at least 4 years. There were some signals of harm, but sort of surprising components of the serious adverse events. And finally, the primary data come from an unpublished study of 156 patients with 1 year of follow-up. So this is a relatively small amount of data for making judgments about the net health clinical benefits. Next slide, please. And because of that, we gave this an ICER rating of P/I, promising but inconclusive. There are some clear benefits. There are concerns about harms, and they're still an evolving evidence base. Next slide, please. So with that, I'm going to leave apitegromab and turn to our second clinical question, which is what's the value of SMN-targeted therapy after gene therapy, in particular adding either nusinersen or risdiplam in patients who've already received gene therapy. For nusinersen, there's one study, the RESPOND study. It's an uncontrolled study of using nusinersen in patients with 2 copies of SMN2. And in the data that are available currently, the report is patients followed for 302 days. They split it into 2 different groups. One group of 21 patients who received nusinersen early, before the age of 9 months. A second group of 13 patients who received nusinersen at 9 months of age or later. And both report an increase in the Hammersmith scale, but it's a subscale used for very young children. So it's a variant of the Hammersmith-2. And in both, they saw a fairly substantial increase of 8.7 points in those who received the therapy before the age of 9 months, and 6.9 points in those who received therapy after 9 months. The study is ongoing. The primary endpoint is after 778 days of follow-up, and that should be completed in October of this year. Next slide, please. For risdiplam, there are 2 observational studies. One, the Jewelfish study, is really a mix of many different patients who've received many different types of therapy who are then receive risdiplam. Among the many patients included in the Jewelfish study, there were 14 patients with 1 to 3 copies of the SMN2 gene who had received gene therapy. And at 104 weeks, they had data on 9 of those 14 patients, and they had an increase of 7 points on the Hammersmith scale. And I think in part because of this, the company has initiated 2 phase 4 studies to study the value of risdiplam in this population in more detail. There's one other study not funded by the company. It reports on 20 patients, again, with 2 or 3 copies of SMN2. Again, you see the theme now that we're identifying patients. So with newborn screening, we no longer call them type 1, 2, 3. We focus on the number of copies of SMN2. And in these 20 patients, they just reported that some patients had improvements in swallowing, some patients had improvements in respiratory function, and all patients had stability or improvement in the Hammersmith scale. So next slide, please. In terms of harms of these 2 therapies, nusinersen primarily has adverse events related to the lumbar puncture, so headache, back pain, vomiting. Risdiplam, on the other hand, has an increase of fever, diarrhea, rashes, and arthralgias compared to control patients, but they're relatively modest. Next slide, please. There are a number of controversies and uncertainties, primarily that these are small studies and uncontrolled. And it's also hard to interpret what happens in these very young people as they grow. They are reaching new developmental milestones just because they're growing. but they're also getting heavier, which means if they've lost motor neurons, they may not be able to manage that excess weight. So it's really hard to assess these gains that we saw. How would they do compared to a placebo group? We have no control group here to really assess this. So it's challenging to interpret these data, though, of course, you saw the gains in Hammersmith here were much more than we saw with apitegromab in that study, but a very different patient population. Next slide, please. So our evidence ratings for nusinersen, we gave it promising but inconclusive, primarily because we saw these relatively large increases in the Hammersmith, but you also have the harms of intrathecal therapy, so lumbar punctures every 4 months, and whether that is a net health benefit in patients who've already received gene therapy is just challenging without controls. For risdiplam, we actually gave it a better evidence rating, C++, mainly because of the difference in the mechanism of delivery. It's an oral therapy, so you don't have the complications of intrathecal delivery of the therapy. And we felt that the evidence for benefits plus very minimal harms, we felt that we could at least say this would be comparable or better, though lots of uncertainty, hence the plus plus. Next slide, please. And finally, I'll turn to the last research question. So what should we be doing as first-line therapy in asymptomatic infants identified at birth through newborn screening? So there are no head-to-head trials of the 3 therapies. All 3 have strong evidence of benefit. So they clearly are keeping children alive who otherwise would die. They are avoiding the need for permanent tracheostomy and being hooked up to ventilators to preserve respiratory function. And they have remarkable evidence of achievement of normal motor milestones compared with historical data. But in the 3 studies, the case series that are available, they have very different study populations. There have been attempts to do network meta-analyses, but they've been heavily criticized because of the differences in the patient population studied. So we didn't attempt to do our own network meta-analysis here. Next slide, please. So I didn't present the data in your in the report and in the tables that we'll refer to with the voting questions. I have a little more data on the outcomes in each of the 3 observational studies, but in all 3 cases, we feel like the evidence is insufficient to say whether risdiplam is better than nusinersen, risdiplam is better than gene therapy, nusinersen is better than gene therapy. All of those comparisons, we really feel like we don't have enough evidence to say that one is clearly superior to the other. Next slide, please. And with that, I think I can take a few questions. Thank you for your attention.
Thank you, Jeff, for the review of the evidence. Before we get too far along, Dr. Crawford is one of our experts, and I wanted to give him a chance to introduce himself and to declare any conflicts of interest before we get to the questions. So, Dr. Crawford.
Very good. Can you hear me?
Yes, sir.
Oh, good. So, Dr. Tice, that was wonderful. I thought that was a really nice summary of things. I'm a child neurologist. I've been involved in SMA things for a long time. Came to Hopkins in 1987 and have been pretty much concentrating on SMA as my primary research element in all of its dimensions, from the genetics and the pathology and the clinical care and the design, the clinical trials, and now the conductance of those clinical trials. And so this is of great interest to me. The only comment I would have on the things from before is that I agree with the controversy about that minimum of 3 Hammersmith points is meaningful. That sort of derives from a more statistical approach to meaningful than a personal standpoint. People with SMA are very stable, and they get to be exceptionally skilled at using the power they have. many people with SMA are in advanced degrees, advanced SMA. They're the ones that do these really inspirational kinds of things with the power that they have. And every one of them says, if I could just do this a little bit more, I could do, and they'll enumerate the number of things they can do. So small bits are very, very meaningful to the people who have the disease.
Thank you. Okay, for questions, I think I saw Don's hand up. Don, did you still have a question? Go ahead.
Yeah, thank you. And appreciate both Dr. Tice and Dr. Crawford with their expertise. So thank you very much. My two questions are related. They're really related to the NMSE, and I'm wondering for either of you, on one hand, if there are any inter-rater reliability studies on the performance of that test, given the fact that it is fairly time-consuming and, relatively speaking, somewhat complex, that you need special training, et cetera. And then on top of that, Dr. Tice, are the deltas of the minimally clinically important differences, are they more profound at lower baselines as opposed to higher baselines?
Well, I'll take the question for me first, but I think then I'll let Dr. Crawford and our other experts address the issues of the validation of the scales. Again, we don't have published data. We requested subgroup analyses from the company. We just don't know whether there were differences there. Potentially, they did. I didn't address this, but they did limit the patient population to a certain range of the Hammersmith scale so that there was room for patients to get better and there was room for patients to get worse. And so that was thought about. I think Dr. Crawford was involved in those decisions about the design, so he may be able to address that a little bit more. But I'm unaware of any data yet on that, though he may be able to comment in general about how patients respond.
So the studies have been, the Hammersmith has been extensively studied, but the problem is we're dealing with kids, and kids have a tendency to say on one or another day, I'm not going to do it, or they may be sick or something. So there's a significant test-retest variability, but the actual performance of the thing is of the test is pretty good and subject to a lot of validation with videos and other sorts of things. So from that standpoint, the other thing is what is the meaningfulness of a 2-point change at the lower end of the scale versus a 2-point change at the higher end of the scale is almost an unanswerable question. We can answer it from the standpoint of statistics, but the meaningfulness is according to very different scales along the way. But I think it's possible to say that individuals will comment about the differences at every level. 2 or 3 points is a lot at every level.
Yeah, I just wondered if the deltas were higher with a lower number is what I'm getting at.
Ooh, I don't know that.
Dr. McMillan, did you have a comment?
Yeah, I just wanted to jump in to add to what Tom's saying. There are papers that are published in the literature looking at the interrelator reliability of the HFMSE, and I can put that in the chat just so you have that reference if you'd like. So that's available, published. The other piece that I wanted to add is I completely agree with the debate around what is the minimal clinically significant change with the Hammersmith. And again, there was a recent publication by Karate, a US and European group that looked at a statistical analysis. They found that looking at type 2 and type 3 SMA, that the MCID in the positive direction was a change of 1.5, and in the negative direction was minus 3.2 to be clinically impactful, clinically significant. And that also was really well grounded with parent and caregiver observations as well. I'll put that reference in the chat as well. Thank you.
I thought of one other thing that wasn't mentioned by Dr. Tice. We designed the apitegromab study on the sitters, the non-walkers. So if you were type 3, you had to decline to the point where you were no longer able to walk. And that's entirely because of clinical design, where our sweet spot of the outcome measures was most likely to be meaningful. We also excluded the Zolgensma kids, mostly because that wasn't available at the time of the initiation of the Scholar Rock program. So there's no plausibility reason why it shouldn't be extended to Zolgensma. We just didn't include them in the initial study.
Thank you for clarifying that. That's important. Rachel?
Thank you. This is a very interesting and informative presentation. One of the questions that's been brought up regards the burden of traveling for IV infusion every 4 weeks. I'm not a clinician. What do we know about what proportion of these patients in the real world, right outside the clinical trial populations, are already on nusinersen, which requires the intrathecal injections every 4 months? Is it possible those can be co-administered in a single visit, recognizing this wouldn't apply to patients on risdiplam?
So nusinersen's every 4 months, and apitegromab is every month.
Right, but in theory, if you're coming,
Sure.
yeah,
Sure.
so it's a partial, right, help.
Yeah, and home infusions will be possible, but if you're outside of a network where they have pediatric nurses for home infusions, that might become a limiting factor.
David?
Yeah, thank you for the great summary. So I'd like to get better understanding about this Sapphire trial result. And then the main benefit seems to be driven by net decline in functions among placebo group, and then placebo group kind of combinations of both nusinersen and then risdiplam. So any ideas about what treatments they actually receive as a standard of care affects the result? Those are my first questions. Second question is age group 2 to 12 are pretty big for me, particularly from child development perspectives, and then how they actually accommodate their functionality. So is there any kind of age dependent response as well? So those are my 2 questions.
So again, I mean, we have the frustration of we asked these questions from the company and didn't get data. And it hasn't been published. I'm hopeful that at least in the supplementary materials, we'll look at some of those subgroups. Was there a difference in patients who received risdiplam as background therapy versus nusinersen? Is there a difference in those ages 2 to 6 versus 6 to 12 or something like that? As far as I know, those haven't been presented yet, so we just don't know.
As to the 2 to 12 thing, the Hammersmith score is you get additional ceiling effects when you develop contractures. And so people who are a little older than 12, many of them get a ceiling effect because they can't unbend their legs anymore, so they can't do the mobility thing. So the 2 to 12 for me is sort of the sweet spot for being able to measure a difference.
Thank you. That's a good point. Reem, and then we'll get to our one of our patient participants or parent participants. Reem.
Right. Thanks. Jeff, thank you for this really well organized presentation of multiple and complex topics. I don't know that maybe the question is a perfect segue for one of our patient representatives, but I have a couple comments that I just wanted to make as I'm thinking through this. The first, Dr. Tice explained this multiple times in his presentation, but I was struck by what's the harms. And despite that this may be a chance, it was just surprising that it was more in the people who are getting treatment. So just kind of want to state that, and I appreciate that you did explain it pretty well. The second point is, while I also understand why an NMA was not performed, in my head, that's what I'm trying to do. I'm comparing them. I'm trying to kind of make sense of all this. I am still doing a much of this comparison between the treatments, which is also quite challenging because not only the population were different, and many of my colleagues earlier really asked about, does this scale perform differently? And that's what we're struggling with and trying to understand. And the last point/question I'm going to say is, do we have or know of any data from the patient and caregivers population about their values regarding a preference, especially for the group who is identified at screening? So we have two groups, one identified on screening and one not. Do we know anything about the preference for, we do one-time treatment versus IV versus intrathecal, for what's going on there? Because that's also an important kind of part of what we're all thinking about. And I understand that these values may be different for somebody who's already developed some, complications or implications of the disease, but that also is helpful to know. Thanks.
Dr. Tice, you want to weigh in?
Well, I can start, but I think I was going to punt it to you. I mean, my sense is that what typically happens now in the majority of patients, but that, again, patient and caregiver values will differ, and so there is some variability, but takes a little bit of time to know that you can do gene therapy. Risdiplam is pretty easy. I think in most cases, people are started on Risdiplam because they can do oral therapy in the kids, and then they test and do they have immunity to the adenovirus vector needed to deliver the therapy. If they have lots of antibodies to the adenovirus vector, the gene therapy won't work. But most of the time, it's okay, and then they need to get insurance approval. But gene therapy is typically delivered within 2 weeks or so. Some variability there, but I think that's the most common pathway, but I think all pathways are possible. But we'll hear from the clinical experts who really know far more than I do.
So humans are complicated, obviously, and take many different paths. The other thing is this, nusinersen was only available the last month of 2016, and really in delivery since 2018, and the other ones got added along the way. So a head-to-head is very difficult because of one thing, that what you start with has a huge influence on what you finish off with, and subtle differences in how much a baby does as a 2-month-old or as a 1-month-old have massive effects later on. So this is the problem of comparing, because each of them started with slightly different groups. The first kids for the Spinraza, for example, if you were thought to be asymptomatic, you were in, and if you were thought to be symptomatic, you were out, which meant that the investigators had a life or death decision. Do we want to include you and save your life, or are we going to exclude you otherwise? So they were very dirty about including weaker individuals just because they were humans. Later studies were more and more strident. And so when you try to compare those beginning groups, it's very, very hard to figure out what their outcomes are going to be later on. I personally don't know that we have any evidence that any one of them doesn't saturate the potential for benefit. And what we're actually seeing is the playing out of the damage that was done before we saw them. Most kids with 2 copies have evidence of a lot of motor neuron loss before birth. And so that has a big factor in what they're going to come. The 3 and 4 copy kids probably are not losing motor neurons until somewhere in the course of their first year. So that's why those ones do so well. I forgot if there was another question. The question of whether there's additional benefit in adding another one is going to involve carefully matching paying for these studies, which none of the companies want to do, and outcomes. I don't think there's any prospect that we're going to have a head-to-head comparison, even in a retrospective way.
Thank you. I wanted to pull in Portia and Giles to see if they had any questions or comments they wanted to make before we move on to the last Stacy's question and then to the pharmacoeconomic presentation. So Portia, you want to?
Thank you. Yeah, first of all, on the patient sort of preference on the method of administration, I would say, so I mean, in the UK, I'm on a lot of WhatsApp groups with lots of families, and this kind of discussion comes up an awful lot about treatment choices. And I think the perspective is that Zolgensma is the gold standard treatment, and they think that the outcomes are the best. And it's not just because it's a one-off. There seems to be some sort of perception that, whether it's Chinese whispers, that Zolgensma has the best outcomes. And I do quite a lot of work trying to reassure families that that's not actually what the trial data shows us. I think that for patients that started on Spinraza when that was the only choice in the UK, so those kids like my son, they're older now, there's a perception that the drugs that go into the intrathecal space are more effective because it feels like to a lay person that that's where the problem is. So if you're putting a drug right there, then that's going to be doing the best job rather than something that you're taking orally. And I kind of put myself in that box, even though I'm, very clued up on all the data. Because my son was so severe when he was treated, I'm very reluctant to switch to the oral treatment because I know that every individual has an individualized response. So I'm not at the moment willing to risk that, if you like. But all these thoughts and perceptions are not necessarily based on facts, I would say. And I think social media has a big role to play in these kinds of conversations and people sharing their very mixed. It's like the side effects with risdiplam. Some people experience real gastric problems, and that can put someone off. But the next person is absolutely fine. So it depends who you're talking to and whose voice whose voice is the loudest. In that sense. So that was that one. And my other question was why are there no type 1s in this apitegromab trial? Was it just to keep the heterogeneity of the data set, or was there any other reasons?
It was driven by the fact that the Topaz study was done way early in the course of things, and it was over my objection. I can tell you that. I think that they grew the fact that they allowed that to pass, but it was just trying to keep, you have a small company with not a lot of money, and you want to ones that can show the benefit, and you pick your sweet spot. And
right,
that was what.
Do you think it'll impact the recommendation for type? Do you think it will have an impact on the recommendations?
To be honest with you, the type 1, type 2, type 3 designation is an antiquated idea, and I would like to get rid of the type 1, type 2, type 3 entirely. I suspect that for example, you're type 2 when I say you are.
Exactly.
So
exactly. I'm always banging the drum, and I don't like the diagram in the table. I like to see it as a spectrum. I think it's much more helpful for everyone to see those overlaps. Like there's so many overlaps between type 1 and type 2. It would be unfair to limit it to any type.
I don't want to argue from plausibility too much because I like data, but let's remember this is a muscle-based therapy and is entirely independent of the copy number, entirely independent of the pathophysiology of motor. It's basically making normal muscle have more contractile protein in it and shouldn't be influenced by any of these other considerations.
Yeah,
thank you.
That will be important for the policy roundtable. Keep that in mind.
Yeah.
Yes, thank you. Dr. McMillan and then Giles, real quickly, we're running over on we need to get to the economics, but we're interested in your comments.
You go ahead, Giles.
It's just a really quick point, and I think it was one that Tom made, was around what is the meaningfulness of some of the Hammersmith points. And I think that it's such a huge question that we come across wherever you are on the journey of your SMA journey. So there's another overlapping part around about everyone's joining at different times, a lot of them. So if it's the 2s and the 3s, if you are asymptomatic or showing symptoms, the effectiveness of that is massively variable. You can go on a very different path. And I think that's something that, as a patient perspective, when people are making those decisions about which treatment to go down or potentially other treatments to go down, that's something that needs to be included. The bit about the meaningfulness around the Hammersmith, and particularly around that kind of 3-point change within a peatograb, that 3 points is so varied again. Whether it could be someone feels that they will be able to stand up with one knee to standing, or it might be the actual difference being able to lift their head off the floor, or going back to Sarah's original comment in her opening presentation around about pulling the duvet up yourself. Like, it's so different to say that 1 point or 3 points could be across that entire spectrum. And I think it's just really around about the individual and the individualization of that choice between whatever people might decide is the most effective, or you could say, is the kind of juice worth the squeeze? Is it worth the IV every 4 weeks and the extra hassle?
Thank you. Dr. McMillan.
Yeah, and then I'll just quickly add, just related to Reem's question and comments, is I think when outcome is evaluated in the pivotal trials of each of the approved drugs, so specifically Nurture, Sprint, and Rainbow Fish, that motor outcome be considered, but also bulbar function be considered separately. With the motor outcomes, there's the duration of the trial, but it's important to also look at the long-term extension studies, because we know that particularly for the 2-copy patients, many of them did walk, but significantly like after the trial had ended. So that's really an important piece. And the bulbar piece is very important as well, in terms of, particularly again for the 2-copy patients, maintaining weight and being able to feed independently without a G-tube. So I think those are indirect comparators, but they're still noteworthy among those 3 pivotal trials. Thank you.
Thank you. At this point, I would like to introduce Linda Liu. She is a researcher from the University of Washington, Department of Pharmacy, and she's going to present the economic model for us. Thank you, Linda.
Thank you so much, Jill. Next slide, please. So first, I'd like to acknowledge the other members of the economic team who contributed significantly to this analysis. So we have Josh Carlson, Woo-Jung Lee, Marie Phillips, and Hui-Shun Chen. Next slide, please. For disclosures, the University of Washington did receive financial support from ICER for work in this assessment. Additionally, Josh Carlson has received consulting fees from Genentech, but not related to work from SMA. And no other members of our team have conflicts to disclose. Next slide, please. So we did not model the other interventions reviewed by the clinical team. The objective of our model was to evaluate the lifetime cost effectiveness of apitegromab added on to the standard of care treatments, nusinersen or risdiplam, compared to standard of care alone for the treatment of non-ambulatory type 2 and 3 SMA in patients aged 2 to 12. Next slide, please. So before we dive into this model, we'd like to highlight the significant unmet need in this population. So the absolute equal value of life year shortfall of 58.2 represents the total health loss patients with this condition are expected to face without new effective treatments. EVLYs assign equal value of life years regardless of a patient's baseline health status, so essentially valuing each year as if it's lived by a healthy individual. The 94.7 proportional shortfall tells us the severity for this condition, that patients lose almost 95% of their potential remaining life years if there were no new treatments. So to put this in perspective, we have listed shortfalls for other conditions, and this unmet need is most comparable to pediatric B-cell ALL, but substantially higher than multiple sclerosis or osteoporosis. So this demonstrates that type 2 and 3 SMA is a condition with one of the most severe unmet medical needs and highlights the importance for new interventions. Next slide, please. So next we're going to move on to our methods. Our model used the standard ICER approach with a co-base case of a healthcare system perspective and a modified societal perspective. And we'll be presenting the results from both of these perspectives today. Next slide, please. So our model followed a simple structure with 3 states and all patients beginning in a sitting health state and are then able to transition to not sitting or death. So we recognize that this may appear overly simplified for this condition, as in reality, patients exist across more granular functional milestones. And we requested milestone-specific data from the SAPPHIRE trial to develop a more detailed model structure, but we didn't receive any. So instead, we adopted 2 key approaches to work within our data constraints while still ensuring we could capture the treatment effect. First, we aggregated those multiple functional states such as sitting with support and sitting independently into the broader sitting state. And second, we use treatment-specific utilities to incorporate the clinical outcomes observed in SAPPHIRE. Next slide, please. So our model cohort was based on pooled data from the SAPPHIRE trial for participants aged 2 to 12, and the table shows their key baseline characteristics. So we like to highlight their mean age of 7.8 years and that patients have been on these standard of care treatments prior to model entry for an average of 5 years on nusinersen and 3 years on risdiplam. Next slide, please. So here are our model's key assumptions. First, all patients began in the sitting state. This reflects SAPPHIRE's requirements for non-ambulatory patients. Additionally, baseline HF-MSE scores fell within the sitting categorization from published literature. Second, we assume no transitions above sitting due to limited data on milestone distribution. We took a more conservative approach, maintaining sitting as the aggregate state as mentioned earlier and capturing clinical differences through utility adjustments rather than as explicit states. Third, treatment effects achieved in SAPPHIRE are maintained throughout the lifetime. So this assumption is consistent with previous SMA economic models given limited durability data. And fourth, disease progression follows a constant rate of HF-MSE decline for both treatments, and this was based on clinical expert opinion. Additionally, we assumed patients receiving apitegromab begin progressing after 4 years, and this assumption was based on TOPAZ trial data showing 4-year stability, and we explored different stability periods in scenario analyses. Finally, we assumed that there were no treatment discontinuation, as none was observed in the trials, and treatment may be helping maintain function even without any additional improvements. Next slide, please. So we incorporated incremental utilities to model clinical outcomes in 2 parts. The first part is patients who achieved the moderate motor function score gains of 3 or more points to their HF-MSE. And then the second part are patients who gained or lost a motor milestone during the trial. So the figure shows a 17.9% difference between the treatment arms in SAPPHIRE for patients achieving this moderate motor function score increase. And within the 17.9% on apitegromab, we assigned 9% as having gained a milestone, and they received a 0.08 utility gain. So we assumed all patients observed with a new milestone in the trial also had the moderate improvement in their HF-MSE score, so that the remaining 8.9% who only had this moderate motor function score gain received a utility increment of 0.06. And then the remaining 82% of patients on apitegromab without either of these gains just maintained baseline utility levels. Additionally, 3% of standard of care patients lost a milestone during the trial, so they received a utility decrement of 0.02. Next slide, please. So combining these increments with baseline utilities from Belter et al. Results in the treatment and state-specific utilities in the table here. The baseline utilities were HUI3 values obtained using the 2019 CEPAC SMA Community Update Survey. Next slide, please. In the modified societal perspective, we incorporated caregiver utilities through an additive approach of 1 caregiver plus 1 patient equals 1 family quality value. Caregiver utilities were derived from Lopez Bastida, which were used in the NICE technology appraisal for nusinersen, and these utilities are displayed in the table below and are patient state dependent. Additionally, we included bereavement disutilities obtained from an unpublished manuscript by Tara Lavelle. These were applied for 20 years post-patient death and consisted of values for less than 5 years, 5 to 10 years, or greater than 10 years post-death. Note that these values were derived from patients with type 1 SMA, which may introduce some uncertainty when applied to our cohort of patients with type 2 and 3 SMA. Next slide, please. Another element of treatment effect is the impact on disease progression. So the figure shows average HF-MSE scores over time in our model. In the first year, following the blue box, we track actual scores from the SAPPHIRE trial. And after that, patients on standard of care in the gray line follow a constant decline, reaching the not sitting threshold of a score of 4 after 10.6 years on average. Apitegromab patients maintained stable scores until 4 years based on the TOPAZ data, then declined at the same rate as standard of care, reaching the not sitting threshold after 14.4 years on average in our base case. So we modeled disease progression assuming that HFMSE scores declined at a parallel rate in both treatments, and that the rate of decline was observed from SAPPHIRE's placebo arm. I'd like to note that there is considerable uncertainty around these progression assumptions, and we varied the stability period for apitegromab in scenario analyses. Next slide, please. So mortality followed the approach from the previous ICER 2019 model, using a separate mortality model for each health state. This, combined with different rates of disease progression between treatments, resulted in differential mortality in our analyses. Additionally, we included pneumonia as a serious adverse event, assuming a 1-month duration, and conducted a scenario analysis excluding it to test model sensitivity. Next slide, please. For treatment costs, we used an annual placeholder price of $350,000 for apitegromab from IPD Analytics. Annual costs for standard of care treatments can be seen in the table, and any additional costs included in our model are detailed in our evidence report. It's important to note that all of our cost effectiveness results are based on this placeholder price, assuming it was the price of the drug. Next slide, please. And moving on to our results. For the healthcare system perspective, total cost for apitegromab plus standard of care is approximately $11 million using this placeholder price, which is around 5.7 million higher than standard of care alone, for a gain of 0.7 quality adjusted life years, 2 equal value of life years, and 1.9 life years. The resulting incremental cost effectiveness ratios was 7.7 million per quality adjusted life year gained and 2.8 million per equal value of life year gained. Next slide, please. So because caregiver effects were substantial in this population, in the modified societal perspective, incorporating these effects, we see larger gains in quality adjusted life years of 2.02 compared to the previous 0.74, and in equal value of life years of 3.03 compared to the 2 seen in the healthcare system perspective. And life years and costs are the same in this perspective as the previous one. So this results in lower ICERs of 2.8 and 1.7 million respectively per quality adjusted life year and equal value of life year gained. Next slide, please. So we varied individual parameters to examine the impact on the ICER and found that the duration of apitegromab is able to prevent disease progression, had the largest impact on results, with the ICER per quality adjusted life year ranging from 2 million to 7.8 million in the modified societal perspective, followed by the dose of apitegromab, the months patients stay stable on standard of care alone, and the baseline utility for type 2 sitters. So this plot shows the results for the modified societal perspective, but similar results were observed in the healthcare system perspective. And in both perspectives, apitegromab with standard of care was not cost effective at any of the common price thresholds across 1,000 PSA simulations. Next slide, please. So when standard of care involves high cost interventions, life-extending treatments may not achieve feasible value-based pricing under conventional cost-effectiveness methods. So as a result, we conducted an ICER reference case where we removed all non-intervention costs as well as costs associated with standard of care treatments to allow apitegromab to achieve value-based pricing. So this scenario resulted in slightly lower ICERs in both perspectives. We also conducted a scenario without pneumonia and found that it had almost no impact on our results in the third row. And then finally, we conducted a scenario for the most optimistic case, where patients experienced no disease progression throughout their lifetime on apitegromab. So although this had a large impact on all ICERs across both perspectives, they all still remained fairly high. Next slide, please. Additionally, we conducted health benefit price benchmarks to calculate the annual price of apitegromab needs to be at to hit common price thresholds of $100,000, $150,000 per quality adjusted life year or equal value of life year, and the associated discount from the annual placeholder price, assuming it was correct. So note that we were unable to achieve any positive price with the base case, and all results reported here are using the ICER reference case, where the only costs remaining in the model are costs associated with administering apitegromab. In this scenario, to reach common price thresholds, discounts of 91% to 96% from the annual placeholder price are required across both perspectives. Next slide, please. So some limitations in our model. One limitation was in the available data. Our analysis relies heavily on the unpublished 12-month SAPPHIRE trial as a primary data source for apitegromab. So a short follow-up period creates significant uncertainty around the treatment effect and its durability. Additionally, SAPPHIRE enrolled a specific patient population, but subgroup data was limited. So we were unable to predict how treatment effects vary across different patient characteristics. Just note that applying our results to populations that differ from the trial cohort requires strong assumptions about treatment effect generalizability. Additionally, we lacked information on the distribution of patients across different motor milestones, as mentioned earlier, which restricted our model to a simple structure. In addition to limited trial data, we also had limited utility data. So trial reported outcomes as changes in HFMSE scores, and translating scores to improvements in meaningful utility changes proved pretty difficult, given most utility data is milestone dependent. Second, while we use established utility instruments like HUI-3 and EQ5D, these may not fully capture the specific impact of SMA on patients' quality of life, though they represent the best available evidence. And there is also substantial uncertainty around disease progression, especially for patients already on SMN therapies with apitegromab added on. So as a result, our model showed life extension. However, there's currently no real-world evidence supporting this outcome. Next slide, please. So stakeholders provided feedback on our draft report regarding model structure and assumptions. We believe that the incremental utilities effectively captures small motor improvements, and we had added a co-base case for family impacts. We modified our base case to include disease progression as requested, and scenario analyses showed pneumonia assumptions had negligible impact on our results. Finally, we used the established 3-point HFMSE threshold that is clinically validated, rather than the 1.5 threshold referenced by the manufacturer, as it wasn't reported in SAPPHIRE, and we also had no utility data to translate smaller gains in motor function into changes in utility. Next slide, please. So in conclusion, apitegromab and standard of care provided modest gains in qualities, but exceed standard cost effectiveness thresholds if at the placeholder price of $350,000 annually. Substantial price reductions exceeding 90%, given the placeholder price is correct, is required even after excluding a majority of costs in both perspectives. It's important to note the substantial uncertainty in our results, given the limited data and the inherent challenges that come with modeling rare diseases. However, our model provides initial cost effectiveness estimates that can be updated as additional evidence becomes available. Next slide, please. And with that, we will be taking any questions.
Linda, thank you for reviewing that with us. It was very informative. I'll start with asking Stacy, what's your question?
Great. Thank you. So this gets to this question of unmet need and also thinking about how we should be considering the population receiving the treatments and the trials relative to like the population who we might have moving forward. So once screening is fully implemented and people are, or children are receiving disease modifying therapy very early on. So I guess when I'm thinking about this and thinking about the unmet need kind of information presented, I'm wondering, is that among populations who were not able to receive disease modifying drugs early? So we're thinking about a group of patients who we may frankly have fewer of because of the success of the disease modifying therapies. Just curious how I should think about that moving forward.
So I believe the unmet need estimates that were shown were based off our cohort in the model. So it's like based off the patients from Sapphire. But if David wants to add to that.
So just going back to what we heard from the clinical experts before, I do think to the extent that we're the studied population, hopefully in the United States over time, new people won't be joining that studied population. But the children who previously would have been type 1, who are born already with some weakness, you could imagine that a strength building therapy, there may be interest in providing that to them, and that isn't who we're modeling here. So it isn't exactly the same situation. But if the question is, gee, do we think apitegromab will get used at all in SMA? I think the answer is probably. But over time, you're right, I think there'll be fewer and fewer. There's a prevalent population of people who, like the people who were studied, and if we're doing the newborn screening well, we won't be seeing people joining that prevalent population.
David, that's exactly correct, but I think I want to correct, this is Tom Crawford. The most common form of SMA is the baby form, that the babies die. And so that's 60% of the population. So we're going to see a lot of 2-copy kids growing older that are in what used to be the 2-copy kids. So I think that's going to be a bigger and bigger portion of things.
Right. What I was trying to suggest, though, is that that population won't really look exactly like the prevalent population that was studied here. Many of those 2-copy children end up ambulatory.
Not as many as was earlier presented. There's going to be a substantial number of non-ambulant 2-copy kids.
Timothy, sorry, I couldn't get my audio up.
Yeah, I didn't know if you was waiting for you to. So first of all, thanks. Very important topic, and I thank our patient experts for being here. And my heart goes out to them on this important consideration, and the clinical experts and the ICER staff for great presentations. I'm going to present a little bit of frustration and struggle here, I guess, with the uncertainty that was brought up. Just to state up front, frustration that we don't know of price, and that we have to go forward without that. So the modeling was done without that. That's not the fault of the ICER staff, but I think it's important that they run the model anyways and present what a cost effectiveness price will be. I want to go back to a comment that David Kim brought up earlier about a lot of these results seem to be driven by sort of, well, maybe I'm overstating that, but I'll state it, by the effect in the placebo category, and the sample seems pretty small. So I'm worried about the uncertainty that's driving some of these results, even though it seems, the results are important and substantial, but, I'm still sort of struck and struggling a bit on that question.
Rachel?
So I'm going to re-ask a version of Stacy's question to make sure that I've understood the responses that we've received from the clinician. So I heard, or I think I heard, that the unmet need analysis was based on SAPPHIRE. And that SAPPHIRE, because it excluded Zolgensma, it included the other SMN2 modifiers, but kind of by definition, patients couldn't have received those until somewhat later on. So there was significant harm already done prior to that. So should I think about the unmet need analysis as being primarily relevant for older patients, including children, but also adults, for whom those technologies just were not available at an important time in their lives, compared to a different population of young children who've received these disease-modifying therapies immediately after birth. Now, I heard both Dr. Rind and Dr. Crawford say that there is expected to be a difference in those populations, but there will still be a significant unmet need, especially among one-copy children due to issues of biology and also timing. Is that maybe an accurate summary of the various answers to Stacy's question, or what have I missed?
I think that's accurate. The only thing I'd say is that one copy children don't always live. So it's 2 copy, I think, is important to focus on. There is a large prevalent population. So the studied population isn't small, even though, as Dr. Crawford was saying, 60% of children being born now with SMA have the severe forms. Because prior to 2016, 2017, they didn't live, there's a prevalent population of the type 2/3s that were the ones studied where there is this unmet need that is very large. That was what Linda was presenting. It sounded like Dr. Crawford felt like I was overestimating how helpful the current therapies are in 2-copy children, and that there's still a big unmet need there. I'm hoping it's not as big as what was seen in Linda's presentation, because we really do see in those children pretty dramatic changes. But perhaps maybe I'm being overly optimistic about what the study results showed for the disease-modifying therapies in 2-copy children.
I think you have it exactly right. The only issue is that the 2-copy kids that are now 3 and 5 years old are they're at a sweet spot where you're not seeing the burden of weakness to the same degree as you will see when they get to school age and other things.
Hugh, would you like to comment on David, piggyback on David for Rachel's answer?
Yeah, no, absolutely. I think to Rachel's point, I think there is an unmet need for all 3 I would say sort of cohorts of children. So one are those children that are identified a bit later in life, that perhaps were not identified on newborn screening and have more significant weakness. Children that are identified on newborn screening and are symptomatic at the time that they're seen and treated. Those are largely the 2-copy patients that we're talking about. And then even the pre-symptomatic, I know people sometimes don't like to use those words, and then are treated pre-symptomatically. As Tom was saying, they can show symptoms later in life. So we know from a lot of these pivotal pre-symptomatic trials that 2/3 or 80% of children will walk that are 2 copies during the time of the trial. If you follow them longer, it goes even higher. And Novartis recently reported their 5-year data where 100% of the children with 2 copies treated pre-symptomatically eventually walked. But walking does not necessarily mean that you're normal. And there are children that are walking that are diagnosed pre-symptomatically that have had motor neuron loss and would benefit from something that potentially would strengthen the muscles that are still connected to motor neurons.
Thank you for that. Okay, we'll move on with David's question.
Yeah, thank you for the great presentations. And I have a questions for modified societal perspective. So I know you incorporated caregiver health gains, but from the broader societal perspective, there are like more to capture as a non-health impact associated with these treatments. And then I'm going to highlight one for the cost related. So I think the treatment time cost is important things to matter in these cases, because epigromab require IV injections every 4 weeks versus the Spinraza was like every 4 months. So basically the time to come to the hospitals to get like going through all the treatments require substantially different time commitment from the different innovations. So that I think that is important part of the societal perspective analysis. The another things in terms of benefits, when we treat like the children, we all thinking about their long-term educational attainment and then productivity benefits. So I know the earlier ICERs for the inherited eye disease, they try to capture these like educational attainments or productivity gains. So whether the UW team actually thought about this, adding this more broader set of non-health impacts into the modified societal perspective analysis.
Yeah, thank you for that. So just, I guess, I'm just gonna start with the education comment. Like, there are a lot of like additional aspects we could look at for the modified societal perspective. We just felt like this apitegromab hasn't shown that it might be impactful in a lot of these aspects. And for the additional costs associated with getting the treatment, I think that is something we could consider adding into the modified societal perspective.
Yeah, I'll just add that, the trajectories here aren't too different on the educational trajectory, so that one wasn't assumed to be a large impact. These patients are also going in a fair bit already, so the actual added increment would not be expected to be too large in terms of the transportation to get the additional administrations either. But it is a fair point that there's probably a small incremental effect there related to the transportation level aspects.
Thank you both. Bijan, you have a question.
Yeah, thank you again. I could appreciate modeling something like this with such a restricted amount of data. I guess my question is about the cost assumptions that you have. So I think for 2 of the SMN targeted therapies, you have gotten the cost from CMS, and I don't know, like these are young patients, right? And I think their coverage would be most likely be coming from commercial payers. So I guess my question is it those 400, or I think both the costs are right now over 400,000. So would that be, I mean, I don't know how much in terms of sensitivity analysis, whether how much lowered, whether you lowered that cost, to what extent, I don't know. And also my other question is more like, because we have folks from the UK, and it would be interesting to understand as to, how these drugs, what's the cost there, what's the, basically how much UK government pays for these 2 drugs versus here in the US.
Josh, would you like to take this one?
So, these costs for this is the standard of care. Obviously, we have a placeholder price for the apitegromab. And this is sort of a background cost we have. They're rather large. There is some variability, but we didn't expect there to be too much variability in terms of commercial versus public payer prices. So these were available prices that we felt were somewhat reasonable. They are varied in sensitivity analysis, and that background cost didn't have large effects, as we saw with that one-way sensitivity analysis approach there. I can't speak for the UK. My guess is that they are less expensive, as we typically see with how NICE evaluates and negotiates their pricing. So my guess is they're substantially lower, but still relatively high. They do have a separate sort of orphan drug carve out, which allows for some of the higher prices here, but I can't speak beyond that.
Okay, Bradley Martin.
Hey, I had a question on this, maybe to the clinicians or maybe to you, Josh or Linda. In terms of the model, I know the Sapphire chart didn't include mortality, but you modeled it. Wanted to get a sense of, you projected a survival in the standard of care arm of 12 years. Is that typical? I know you've I read the section on validation, but I just want to know if that was an expected survival benefit given this patient population that was enrolled in the trial. And if clinicians have a thought on that, I'm just trying to get a sense of the model validity of that kind of
The
overall.
survival benefit is longer than that. That was time. The initial one on the chart that you saw was time to not fitting, which was around that. So the actual survival time is longer. And also these are discounted values, meaning that the actual projected survival without taking into discounting, which is used to basically adjust for time preferences. So the actual life extension is longer than that. I don't have the exact figure, but we did look at that sort of relative to what an expectation would be, and it did seem to be a reasonable projection.
Okay, thank you.
Okay, so it looks like we don't have any more questions at this moment. So I think we'll move on to the public comments.